Go, go, go. All right. Hi, everyone. Um, I like to start with a slide to sort of get everyone in the headspace that I'm in when I'm thinking about small animals. So we've got Ant-Man riding a tiny worm. When I think about animals, they look a little different to me. I work on meiofauna, which is not a phylogenetic group or anything like that. It's a size class of animals, and they're usually the size that would burrow or swim in interstitial spaces between sand grains. So they're quite small. Many of them are familiar to us, so if you work with C. elegans or know about it, they're like nematodes, like what you see up here. And of course, everyone knows tardigrades, they're really cute, but there's also groups like annelids where we know larger worms that have meiofaunal representatives. But there are groups up here you probably are less familiar with, like gnathostomulids up on the top right, gastrotrix, and acials. But these tiny animals are major proponents of metazoans, so especially in Bilateria, a lot of our extant and successful taxa or groups are within Bilateria, which I can show here. And what I've shown here in blue are all the major groups that have meiofaunal representatives, but in pink we have groups that are exclusively meiofaunal, so we only have microscopic animals in those groups. Just today, I'm going to be talking mostly about this one group here, and what's interesting about it is sister to all the other bilaterian taxa, so you can see it branching off over there. So what are acials? So acials are long thought to be flatworms, kind of related to the free-living relatives of cestodes and trematodes, like those parasites. But as I mentioned, they're now presented as a sister to other bilaterians. There are about 400 nominal species, and they're quite small, anywhere between 0.2 to 10 millimeters. But I'm going to see if I can pull this off and show you this video of a giant. Can you see it? No, you can't. Oh, okay. This is a giant 1 centimeter acial. I like to show videos of it because as beautiful as these microscopy images are, you can't really understand why I love them because they're fat and cute. Oh no, how do I get rid of it? Okay. So they're primarily marine, and they're found in most, oh my goodness. They're found in most places, and it's not abnormal that a lot of the diversity is well known in Europe, especially in Sweden and in Italy. We have a couple taxa that are been reported here in North America, but as you can see, it's pretty abysmal in Canada. There are 0 reported instances of acials. And that's not because they're not here or anything like that. They're quite abundant. So this is one of these intertidal mudflats that I sample regularly. And depending on the season and sample, I can get them in pretty abundant numbers, as shown in these pictures. So I like to think of acials as sort of an early stage research organism. While there are some species that are model organisms for individual research programs, they're still not really well known, and there's not a whole lot of infrastructure for studying them yet. Sorry, I lost my timer. So acials there's some cool things about them. Oh my goodness, that's not right at all. How do I get rid of it? I'm so sorry. There we go. Nope. Uh-oh. How do I? Just close it, right? Perfect. There's the good and bad. So acials are positioned in the tree to be quite informative for the bilaterian ancestor because they're sister to all other bilaterian taxa. We can kind of we can think about what early bilaterian evolution looks like with them. And because they sort of arose in deep time, having an ancestrally cosmopolitan distribution is sort of expected and also good because you can find them everywhere. And for me specifically, their abundance in Canada is great. Now, there are some bad things. They're still undersampled. So the amount of information out there is not great. And maybe the reason for this is because they're small, they're really simple, they explode if you look at them wrong. Their features are delicate, sometimes even transient, and worse, invisible, and then they're not preservable. And for molecular data, they're really, really small, so it was kind of hard to get started with sequencing them, but it's getting better with technology improving. But existing literature is pretty dated. A lot of the stuff we have to work with is low quality images, maybe a line drawing from the 60s. and oftentimes just no molecular data. But an example I like to give of it is that there are some known ACL species in the Northeast Pacific. A lot of the time I have to work with line drawings or really bad like Xerox copy quality pictures. And so I was told that something as simple as colored images would be considered a technological advancement. And I'm a giver, so I did it. I took colored pictures. And I know that I kind of knocked the line art here, but it's enough for you to still identify species to at least being something that resembled what was described in, say, the 60s, or maybe sometimes it's the 1860s. But with DNA, I've been able to parse out potentially new species using 18S for now, but I've got a couple other barcodes in the works with the biogeography, and for even some of them, we found morphological differences separating them from congeners. Even like right now, really conservatively, there's 8 undescribed species from one site. And so what's cool is on this tree on the left, it's not super high resolution, but you can see there's 8 species that are scattered all across Acela. And so the presence of different ACL groups locally has opened up the possibility of studying trait evolution across the entirety of ACLs. And it also showcases phenotypes that can inform us of ancestral states when we're trying to understand how early bilaterian evolution occurred. So I just want to show the major groups of ACLs right now. The black stars are groups that I have representative species of. Now, I couldn't describe synapomorphies for these groups even if I wanted to, because they're primarily molecular, and that's just the state of the systematics right now. But I like to note that the thickness of the bars here show the relative species diversity in each group, the largest one being convolutids at the top that have 119 species. And you'll notice that all of that diversity is concentrated in this one group. It's known as a fast-evolving clade named Crucimusculata. And as I said, the synapomorphies in ACLs are usually molecular, but it's really nice that in this one, they're actually accessible to us. They're named after crossover musculature, Crucimusculata. And so on the left there, you see an example where there's the muscles crossing over, and then the one on the right is an outgroup that doesn't have it. It's just like parallel muscles. Oh gosh, do I try and show you a video, or is this gonna suck? That's okay. Just believe me, it's a cool video. So because these are simple taxa and it can be difficult to observe their features, you need to apply a host of imaging techniques to really observe them. Just for this talk, though, I'll only be giving you examples of these two. I've shown you the color microscopy images already, but now I'm going to show you some preliminary data I've got from muscle staining using fluorescence imaging. I like to start with this example because you have this juvenile that's trait barren, but when you actually stain their musculature, you can see a lot more. In orange, I have nuclei, but what I'd focus on is these strands. In blue, we have the more ventral side of the image, and then in pink, more dorsal, and we've got the crossing over of the muscles. It's kind of hard to see because of the nuclei, so in this example, I show one without the nuclei. Even in a mature specimen where you can already see cool features like these 2 eggs and the ovaries along their sides, when you do muscle staining, you see this gaping hole, which is their massive mouth, which would be basically invisible if I didn't stain it. And so in those 2 examples, I've shown ACLs that are within the in-group Crucimusculata with the crossing muscles. But of course, to test this, I examined an out-group, and this is that weird longworm at the bottom, Paratomella, whose muscle stains have not been published before. Once again, we expect it to look more like the image on the right, where there's no crossing of the muscles. And it's a really long worm, so I went to the back end, and well, there's just no crossing over of the muscles. So it's a nice example where you can use microscopy and see that the synapomorphs do hold true, at least for these 3 worms. So this is just one preliminary example where I can start to study trait evolution in ACLs because I have access to distant ACL lineages and advanced microscopy. I'll be imaging more ACLs to make this a more robust test, but in the future, I would like to use this kind of fluorescence imaging to label other tissues of interest. The biggest project that I have in the works right now is I'm testing nervous system staining in non-model organisms, such as these ACLs, but other megafauna as well. But in this particular radiation Crucimusculata, they allegedly have really diverse nervous systems. It's not all published, so it's kind of like word of mouth. So I'd like to make it real and actually take the images and publish them. And of course, being sister to bilateria, it's kind of interesting to study the centralization of the nervous system in a group that is not like the rest of bilateria. So lots of upcoming work in imaging, like the nervous system staining. I'm also interested in 3D reconstructions And there's a lot of molecular work that I just didn't talk about, such as just making more barcoding of phylogenies, as well as metabarcoding and trying to find hotspots of small animal diversity. But in case that's of interest to anyone, I would love to talk. With that, thanks for sitting through my glowy worm presentation. And although I've just gotten started, I have a host of mentors and friends and labs to thank for supporting my work. That's really fast. But.
My name is Theo. I'm a PhD student at Vanderbilt University, and my talk today is about evolutionary dynamics in gestation length in eutherian mammals. So, first of all, I want to thank my, of course, my lab mate, my thesis committee, all my funding resources, and of course, the evolutionary studies. You should go check the booth downstairs. I think we have some swag left. But anyway, we're going to switch gears a little bit. And as I said, my talk today is about eutherian mammals. So no monotremes, no marsupials. And this is because we don't have so much variation in gestation length in these two groups. So why do I care about gestation length? Well, first of all, it's a very important trait, crucial for reproductive success and offspring survival. You can see here in this graph in humans that in x-axis there is a 2-week window, which is crucial for the success of the delivery of the birth. And you can clearly see that the neonatal deaths drop dramatically, that are representing from the solid line, drop dramatically during this window. So that prompted me to explore this trait more. At the same time, we can see that by this very sophisticated figure here, we have huge variation of gestation length from the small mouse to from 20 days to the elephant, which is 2.5 years. So those two reasons made me explore and study this trait. So first of all, I collected data for all the eutherian mammals that are available out there, publicly available, and I employed a Bayesian framework to trace back all the changes that might happen across the mammalian phylogeny. So these circles here, the pink and the blue ones, represent changes that happened in gestation length. The pink ones show increase in gestation length, and the blue ones show decrease in the gestation length. So let's break down this tree more a little bit. So we can see that in artiodactyls we have a fairly flat landscape, not too many changes. On the other hand, we can see changes both either increase or decrease in carnivores. We have some changes in perissodactyls, Uli, Podifla, Pilosa, and so on. And we have a mix of changes in the most speciose order of mammals, rodents. So that made me, there are many questions that came up after this. Given the variation and given all these life history studies that are out there, made me to explore and delve into this trait more. But so far there are many studies that try to explain this variation using just body mass and longevity, and using just a very, very basic allometry analysis. There are two main issues actually with this kind of analysis. First of all, we collapse all the variation, grouping all the mammals together, we collapse all the variation that might exist within the groups. We treat carnivores as artiodactyls and artiodactyls as rodents, and we all know that the life histories are very different. At the same time, there are more actually traits beyond body mass out there. We have female maturity, we have longevity, we have generation length. So my next step was like, what is going after body mass? What is going on with all the other traits? So I started employing different modern methods to explore gestation length and what predicts gestation length, what predicts the variation of gestation length. And the first method that I employed is random forest. I did feature prediction with random forest, treating traits like features. The second analysis that I'm going to show you later, it's the path analysis. It's a causality analysis. It's, well, actually, it's a confirmatory path analysis. We have a set of different models that we believe explain the variation in all their interconnected associations among the traits. After accounting for phylogeny, of course. And the third analysis is the evolutionary correlation. So I wanted to explore all the pairwise correlations of all these traits at the same time and see how they evolve. So here you can see this lollipop plot for eutherians, all the eutherians. I collected data for almost 3,000 eutherians. And we can clearly see that the three most predictive traits based on the trait importance in the y-axis, it's generation length, litter size, and brain mass. And then, unexpectedly, we have body mass, interval, interbirth interval, weaning age, longevity, and female maturity. At the same time, I ran the same analysis to each of the orders, carnivores, Eulipotifla, and all the other orders. I have the slides later. If you're interested, we can talk about that. But the layout doesn't change. Body mass is one of the less predictive traits, is in the middle, and we have brain mass and generation length and litter size, the three most predictive traits. There is only one group that the body mass is the most predictive trait, and it's carnivores. And I think this is associated with Pinnipedia because I exclude Pinnipedia when I run the analysis in carnivores. When I include them, the layout changes there too. So the second analysis, it was the phylopath analysis. Again, this graph, it's in Eutheria. The black arrows here show positive causality between the traits. The blue arrows show negative causality. And the thickness of the arrows is the correlation coefficient that shows the significant associations. And the dashed arrow from the litter size to the weaning age is non-significant association. Again, I've run this analysis to each of the orders, and the take-home message here is that there is no any key trait or major trait that drives the variation in gestation length. Interestingly, the association between body mass and gestation length is weak, but on the other side, we have two interesting associations between gestation length and brain mass. Gestation length drives in a positive way brain mass, and gestation drives in a negative way litter size. And lastly, we have the coevolution of all these traits. Indicatively, I present here gestation length, brain mass, weaning age, and litter size. On the positive side, the red box plot here, we have the positive correlation between gestation length and brain mass, which is kind of expected as far as the previous analysis. On the negative side, we have gestation length and litter size, which is in concordance with all the previous two analyses. And in the middle, we have weaning age and body mass. So the major conclusions here are that we should treat mammals separately, and we have to explore more the variation of all these life history traits and go beyond body mass and all the classic traits that exist out there so far. Gestation length is highly correlated with brain mass rather than body mass, and also litter size. And that's actually very interesting, and that indicates the significance actually of the neurological development in mammals, and of course potentially resulting in smaller litter sizes. And lastly, we have the differences in the coevolutionary rates that might be the different selective pressures that act on the traits in mammals. So my future goals is like to explore more this trait using fossil data from extinct species. And my last chapter of my PhD is going to be the genomic underpinnings of that governs the variation of gestation length in eutherian mammals. And that's all I have for you today. You can scan it's online on bioRxiv, most of my research I showed you today. and I'm happy to take any questions.
We do anticipate that it's very interesting in terms of finding these different correlations than one might have expected. Would you expect that the patterns that you documented in mammals would likely also be the case in other major groups of vertebrates, say fish or amphibians, reptiles? You have to guess?
Maybe yes. Yeah. Yeah, I didn't know. I haven't seen that, but I'm happy to explore that to other groups too. It's really interesting because we include more traits and we see the network like as whole. Again, it's a little bit biased because it's like the model. It's a confirmatory analysis. As I said, it's like the models that we believe that explain the biology. At the same time, all the traits and all the associations we set up as a model should make sense. I cannot, like mathematically, can litter size affect longevity, but in biology it doesn't make any sense. Like, how can you associate litter size with longevity, let's say?
How do you infer gestation length from the fossil record?
That's a great question. Short answer, from teeth. You can vaporize the tooth, and they have very specific chemicals and compounds that you can infer the gestation length. Well, actually, when the mother starts the gestation, all the compounds that the baby needs go away from the tooth, and it comes back when after the delivery. So you can infer the birth line.
Cool, thank you.
Of course. No questions. Okay. Thank you.
Good morning, everyone. It's Dylan here from Arizona State University. Today I'm going to talk about the evolution of cell size and growth in prokaryotes. The first thing I want to do is I want to thank Dr. Kerry Geiler-Zamarati and Dr. Michael Lynch for spending the time with me to discuss, this topic. I would say in the last 6 months, probably. So this investigation derived from an evolutionary cell biology class that Michael Lynch teaches at ASU. Anyways, here's going to be the outline I'm going to follow today. So basically, I'm going to be providing a background on the relationship between growth and size. Specifically, I'm going to be talking about theoretical predictions, and I'm going to be showing you some empirical support. Also, I want to talk a little bit about the data collection. Specifically, I'm going to talk about the variables I'm going to use to, look at this relationship between size and growth. And lastly, I'm going to show you some striking observations, and hopefully this is going to stimulate some discussion by the end of this talk. All right, so let's talk about the evolution of growth and size across the tree of life, right? So a long-standing prediction of life history theory suggests that large organisms grow more slowly than small organisms, right? And it is often thought that the mechanisms driving this relationship is metabolism, because it determines the rate at which organisms transform energy into biological work and growth. And this is an intuitive prediction because I think we all agree that a population of an elephant or populations of elephants grow more slowly than populations of mice. So here's some evidence consistent with that prediction. So if we focus on the panel on the left, what I'm showing you here is, see what we have here. On the x-axis, we have the mass at maturity, and on the y-axis, we have the maximum exponential growth rate. And these are all unicellular eukaryotes and some prokaryotes, right? So the general trend is that as organisms become bigger, they grow more slowly. Exactly what the theory predicts, right? But now there's something to point out here. If we look at the upper left corner, we see that the black circles show the opposite pattern. As organisms become bigger, they grow faster, right? So this is interesting. The panel on the right basically shows the same, so I'm not talking about that right now. It's not important. So here's more evidence of that pattern, right? Again, on the x-axis here, we have cell volume, and on the y-axis, we have the intrinsic growth rate. And this is an E. coli, by the way. So again, as organisms become bigger or larger, they seem to grow faster. So now I'm confused, right? Because prokaryotes seem to defy what the theory says. So I want to further, look at these relationships, see what's going on. And the way I'm going to do that is the following. So I collected data on primary and secondary resources, such as these bacteria archaea traits, which is a database with an overwhelming amount of data, right? So specifically to look at the relationship between growth and size, here are the variables I'm gonna use this time. So for growth, I'm gonna use a minimum doubling time. And according to allometric relationships, cell volume seems to be the most appropriate variable to talk about size. However, data of cell volume out there is really scarce currently. The vast majority of the data seem to consist of cell diameter. Now, there's something important to point out here. If you plot cell volume as a function of cell diameter, you can actually tell that cell diameter predicts cell volume really well with a correlation of 80%, right? As you can see on the right panel. So I would say that using cell diameter would be probably as accurate as using cell volume when it comes to talking about size in prokaryotes. So let's see what happens when we look at the relationship between growth and size in prokaryotes. So when we have cell volume on the x-axis and doubling time on the y-axis, here's what we get. As organisms become larger, they seem to grow faster. Just to clarify one thing here, remember, on the y-axis, we have doubling time. This is tricky. That means that as you go down, that's a shorter time, meaning that they grow faster. And the opposite occurs if you go up, just to clarify that. So this is what Michael Lynch and Marshall just showed. It's just, confirming what they showed. However, when you use cell diameter on the x-axis and doubling time on the y-axis, this pattern suddenly disappears. So prokaryotes no longer defy what the theory says. Neither do they confirm what the theory says, though. So that's interesting. But when I started talking about this relationship, I mentioned that metabolism is the mechanisms driving the evolution of this relationship, right? So why don't we look at the relationship between metabolism and growth? Maybe that can tell us something. So to do that, I'm gonna look at the metabolism of protein synthesis, which is controlled by the translation machinery, here referred to as the number of ribosomal RNA genes and the number of transfer RNA genes, right? So let's see what happens when we look at the translation machinery as a function of growth, right? So here we have doubling time on the x-axis and the ribosomal RNA genes on the y-axis. And what we see is that as organisms grow more slowly, the number of rRNA genes seems to decrease. But this decrease can be compensated by the number of transfer RNA genes. So perhaps the number of rRNA genes is not a good proxy for cell growth, as many people think, right? I would suggest, according to my results, using the number of transfer RNA genes. But what do you think it happens when we include temperature in the analysis. Now, you might be thinking, well, now are you gonna talk about temperature? Why? So it turns out that metabolism cannot be seen isolated from temperature. It is highly dependent to temperature, right? Why? Because the speed of a reaction normally increases with, temperature. So I also want to mention the following. If you're going to pay attention once to my presentation, please make that time now, because I think this is the most important point of my investigation. So it turns out that all of the work that has been done about the relationship between size and growth, the authors, they seem to correct growth at a specific random temperature. It is normally 20 degrees Celsius, and they don't provide a reason as to why they do that. So the work you saw from Lynch and Marshall, they normally do that, and there's no explanation for it. And this is very, very problematic because no two organisms have the same thermal restrictions. So let's imagine we have an archaea. Those are usually thermophiles. They can tolerate temperatures of 90 degrees Celsius. And now let's compare that with a bacteria with an ability to tolerate from 20 to 40 degrees. If you bring the temperature down to 20 degrees, obviously the performance of an archaea is going to decrease drastically compared to the performance of a bacteria, right? So here's a better approach, in my opinion. Many people don't think this is acceptable. So I would like to rely on the concept of the thermal performance curve, which can be defined by three basic parameters: the lower bound, the upper bound, and the optimum temperature. So why don't we look at these parameters against, growth and size in prokaryotes? I believe they can tell us, a more meaningful message. So here's what happens when organisms grow at temperatures below the optimum. Apparently, there is no relationship between size and temperature. Now, here is what happens when organisms grow at the optimum temperature. There is also no relationship between cell diameter and temperature. Now, when organisms grow at temperatures above the optimum, we can see that, cells are larger as temperature increases. So this is very interesting. Now let's see what happens when we see growth. Now, for organisms that grow at temperatures below the optimum, they grow faster as temperature increases. The same pattern can be seen if we analyze the optimum temperature, and same can be said when temperatures are above the optimum. So probably growth is not too sensitive to temperature in this case, but cell size is, as I just showed you previously. Now, if that is really true, my expectation would be that archaea would be bigger or larger than bacteria. Now, why? Because we now know that across the evolutionary history of archaea, they've evolved tolerance to high temperatures. If that is correct, then they should be bigger. That's in my opinion. Well, if we look at the relationship or the difference in growth and size between archaea and bacteria, by the way, I forgot to include the p-values here, but now you're going to have to trust me, please. So on the left panel, we're looking at growth. There's no difference in between archaea and bacteria, statistical difference, I would say. But when we look at panel B, bacteria seem to be smaller than archaea, and the difference here is statistically significant, right? Which which is consistent with, my predictions. So that's pretty much all I have for today. If you have any questions, I'll be happy to answer those. Yes.
Thanks for, it's on, apparently not on. Oh, wait, switch. Hear me now?
Oh,
Yes.
yes.
Thanks for a really cool talk. I was curious, in your last slide, you were showing the archaea bacteria comparison, and you have cell size. Was that diameter or was that volume?
Oh,
in this
diameter,
case?
yeah.
It was
This
diameter
is all
because
about
you'd shown that
diameter.
diameter was important
Yeah.
earlier. Cool.
Yeah.
Thank you very much.
Thank you.